So now we're going to solve some of the exercises that are posted on Moodle. So here you can find the exercises if you want to take a small quiz for practice to study for your test. Okay, this is a very easy question, and I'm asking you about a major difference between bacterial chromosome and eukaryotic chromosomes. So look, give a read, and in the next slide, I will give you the answer. So the answer here, very easy. While prokaryotes have a small DNA molecule, so they will have circular DNA. The eukaryotic have a much longer amount of DNA, so they have to pack it using, as we said before, histones, and we will have this nucleosome that is the unit of this packing, and later you will end up with this highly coiled structure that are the chromosomes, but this one are a linear structure. So the answer will be that the bacteria have a singular circular chromosome while eukaryotes have several linear chromosomes. So that is the answer. B. There is no difference between bacterial and eukaryotic chromosomes. That is wrong, as we just said. C. Bacterial chromosome have much more protein associated with DNA than eukaryotic. So that is wrong. Actually, the eukaryotes to pack it, they have much more protein. So C is wrong. Eukaryotic have a singular chromosome, whereas bacteria have several linear. So this is the opposite of A. So it's wrong. And E, the DNA of a bacterial chromosome has a slight different structure. It's not a slight difference, and it's not describing the difference. So this is just not complete, and A is better. So now this is a very interesting question in terms of evolution. I'm asking you how often we find an error. So what is the error rate in the completed DNA molecule? So these are the options, and in the next slide I will show you the answer. So the answer is here. So there is a very slight chance to make a mistake. So it is very interesting because at the end, actually, although there are very few cases, as there is so many events of replication. So for example, in the case of the intestines in your guts, there is a group of cells, a tissue called epithelia, that reproduce every few hours. So there is a very small chance that during your lifetime you will start accumulating mutations. So those small chances, when it's 60 or 70 years old, you start accumulating a lot of mutation, and this is why aging occur and also cancer occur. So the answer will be A. So here I'm asking you about the description of the DNA replication, and here I gave you few statements, so you have to tell me which is the most accurate to describe a replication. So please read it and try to discuss with some of your friends or try to think about it. And as a clue, if you remember, during the video lecture we talk about this group of proteins, this replication complex formed by all these proteins, and the idea right now is that you think about which is the first step, what is the second step, and in particular, to answer that question, you have to remember that the DNA need helicases to separate this double strand, and later the DNA polymerase are the one that synthesize the new strands. So based on that, the best answer will be A. Why? Because the helicase separate the two strands and the DNA polymerases construct the two strands, so this is the right answer. Here, ligases don't separate the two strands, they join strands, so this is wrong. Now, the two strands separate, they don't separate by themselves. You will have to increase the temperature, if you remember during PCR, to separate them we denaturated, so we need to increase the temperature. If you have a DNA molecule that have these hydrogen bonds, to break these hydrogen bonds, you need to increase the temperature, and in that way the DNA separates. So C is wrong, B is wrong. Here the two strands separate, restriction enzymes cut up one strand, so this is what happens when you are doing molecular biology, that you use restriction enzymes, as I said before, that allow you to cut in a specific way, that they act as scissors, and you can insert and then use the ligate to insert a new sequence, but this is not what happened during replication. And something, so this is wrong, and ligase assemble into single stranded code. This doesn't make any sense. So the only potential answer is A, as we said. So this is a question that generally we talk a lot about the DNA polymerase, and that the DNA polymerase add nucleotides in only a single way, independent if it's a leading or the lagging strand. So these are the options. And remember, study what is a 3' and a 5' to answer this question. So to answer this question, if you remember, this is the 5' and here is the 5', and this is the 3' and this is the 3'. And as we said before, the DNA polymerase only add nucleotides in the 3' end of the growing strand, independent if it is the leading or the lagging strand. So, so here it goes from here, from the 3' here to 5' here, and here also it goes from 3'. This will be the 3'. And this will be the 5' as you can see here. So only one option is correct. So in this schematic you can see first the new strand, and here you can see the template strand, and here in this first figure you can see how the nucleotides are there, the nucleoside triphosphate, and how the DNA polymerase attach the new nucleotide into the new template strand. So here they are not attached, and if you see here later, the DNA polymerase release this inorganic phosphate and allow this to attach to each other, to the, in this case, the A to the T, and if you see, they always occur here in the 3' section. So always has to be 3'. So the only answer is A, 3' and 3' end, because as we said, the polymerase always do it in the same way. So this one that are different is wrong. This one that is 5', although they are the same, they actually are attached in the wrong way. And this one doesn't make sense because this is attaching to a different part of the DNA. So A is the right answer. Now I'm asking you about these 5' to 3' sections, and I want that you make the complementary base sequence. So remember, this is fundamental, and remember that here you have 3' and 5'. Okay, so try to see what is the right sequence. So remember that A's always go with T and G's always go with C. So the right answer will be the A will produce a T, the T will produce an A, and in that way. So the answer will be here, and it will go 3 prime to 5 prime here. So the answer will be this one. Okay, the role of DNA polymerase in DNA replication is so very easy. Just have to read these potential options, and I will give the answer in the next slide. So just to remind you, the enzymes called DNA polymerase catalyze the new DNA, adding nucleotides again only in the 3N. And here you already saw this schematic. I just want to tell you that we are trying to explain this in a very simple way, and we are explaining based on E. coli, that is a unicellular organism that is relatively simple. And here we generally talk about the DNA polymerase 3 and the DNA polymerase 1. And however, in eukaryotic, you can have 11, and they're always finding more proteins, more enzymes involved in replication. So this is just a simple way to study it, but they could be more polymerase. So here, as we said, the answer will be A, because attach few nucleotides to the new DNA strands. Separate the two strands will be another enzyme, that it will be the helicase, link together short DNA strands will be the ligase, and synthesize an RNA primer is the primase. So the only option is A.